Morphology and anatomy of Lycopodium stem, that is the supply specimen. So here the aim of the experiment is to study the morphology and anatomy of the supply specimen, that is the Lycopodium stem and Lycopodium leaf. As we just discussed earlier, Lycopodium has different species distributed throughout the world, and these species are we got the bottle specimens which are being sold by different suppliers, and we bought the bottle specimens for our routine work, that is section of the stem as well as leaf, as that is the study of that specimen. For the purpose of the anatomical study as well as for the morphological studies, we need different lab materials for our routine work, that is needle, plate, safranin, methyl green, microscopic slide, microscope, cover slip, pen, paper, plant specimen, etc. So we have to organize, we have to manage all these things before go for the practical. For the study of morphological behavior of the plants, we have to observe the habit of the plant, that is in which habitat the plant is growing, and to note down the characteristic feature of the stem, rhizome, as well as the leaves. And we have also to observe the reproductive structures, that is the cones are there or not in the supplied specimen. For anatomical purposes, for anatomical studies, a very thin transverse section of each specimen, that is, each specimen is the stem and leaf specimen is taken separately, and each of them is transferred through the double staining process and observed under. We have to observe under compound microscope and note down their anatomical characters, which we have seen under the microscope. For the anatomical study, we have used double staining process, in which we have used forceps gently to transfer the section in small quantities, as well as immediately a drop of safranin into the section, and it is set for 3 minutes with the safranin in the section. Then after this, the safranin, we have plot the safranin and with the help of filter paper, and immediately add 70% or 75% ethanol, and let the section be there in the 70% ethanol for 1 minute. And after that, the section is the ethanol is removed with the help of blotting paper, and again another 70% ethanol solution is added, and keep it for 1 to 2 minutes of time. After that, we have to remove the 75% ethanol with the help of blotting paper, and immediately methyl green is added, and it is the section is will have to be kept in the methyl green for 1 minute. And immediately after that, we have to add 95% ethanol and allow the section to sit there for another 1 minute. And then we have to remove the ethanol part, and again we have to add 95% ethanol, and we have to keep it for 1 to 2 minutes. And we have to take care of the take care that the ethanol does not remain so long in the to the samples, because it would appear translucent to blue. Okay, if it is appear red, then the section stain has not been effectively removed. So the double staining procedure should be well organized, and after that gently transfer the section to a microscopic slide with a filter paper or blot paper, and with the help of blotting paper remove the excess amount of ethanol from the section.